Hey Guru Nation, thank you so much for watching. Make sure you like, subscribe, comment, share. It really does mean a lot to me. Today I'm going to make to keep with the theme of the latest videos I did. The last video I did was the comprehensive guide to a screening visit. And today I'm going to do what would naturally make sense. The comprehensive guide to a randomization or a baseline visit. So this one's a little simpler, but when you're actually doing the visit, I would say it's about as complex as the screening visit, maybe even more complex. So although this is going to probably be a shorter video, just know that the length of this video does not equal the complexity of the actual visit when you do it, and there is no substitute for actually doing it. There is also no substitute for following the protocol. I should put this disclaimer here: whenever in doubt, ask your CRA for advice on the baseline visit. So this is obviously for the research sites, but I know a lot of people that want to learn more about research, CRAs and whatnot, they're going to watch too. So first, what is a baseline visit? Baseline visit, randomization visit, they're synonymous. Usually it's visit 2 of a study, although every protocol is designed differently, but visit 1 is generally screening and visit 2 is generally baseline or randomization. So this visit is typically where patients are officially enrolled in the study. And you might be saying, well, weren't they enrolled in screening? Yes, they were enrolled in the study in screening. They were registered as a screen. But in baseline or randomization or visit 2, they are formally enrolled in the study, meaning they are actually assigned investigational product. They're actually assigned a treatment arm, whether it's placebo, whether it's open label, whether it's standard of care. They're still assigned, they're still enrolled in the study because they're given investigational product. So there is no study without an investigational product for the most part. I think maybe for every study there's no such thing, but I always say for the most part just to be safe. So there is no study without testing something. And it's usually an investigational product. So at baseline / randomization / visit 2, I'm going to use all 3 of those interchangeably now. Just know they mean the same thing more or less. You're ensuring that the subject, before you actually enroll them, before you actually randomize them in the study, or before you enroll them in the study, you're making sure that they actually qualify for the study. If you learn nothing else from this video, know this: the biggest mistake you can make as a site and as a coordinator is enroll someone who does not qualify for the study. That's not only a data integrity issue, but that's a potential safety issue for the patient, sometimes a big one. So that's huge. Essentially, randomization visit, baseline visit, it's the same as screening, other than assigning the investigational product. So whatever you did at screening, most likely you're going to do at baseline or randomization. There's of course always exceptions. Check with your protocol, but it makes sense, right? Screening, you're checking if they qualify. Baseline, you're also checking if they qualify, but you have 2 data sets, right? So you got labs at screening, now you're going to get labs at baseline. Why do they do this? The screening labs are usually safety labs. So the screening labs just ensure that there's nothing funny going on with the patient that should prohibit them from enrolling in the study. At baseline, the purpose of the labs, of course it's safety, but you already know the safety more or less because the screening for the most part was not that long ago, right? The screening visit could be a few days from the baseline, sometimes as short as 1 day or sometimes as short as same day, but it could be as long as 2 months, 3 months. Most of the time it's within 2 weeks to 4 weeks of screening is when you do the baseline or randomization. So again, screening labs are to ensure that there's no abnormal lab values for the patients. Labs at baseline are to get a baseline reading before the patient takes the investigational product. So that's really the purpose of the labs here. Same thing with the ECG, same thing with any other assessments. You are getting as recent data as you can prior to the patient taking the investigational product of their assessments. Labs, right, their results, labs, EKGs, and any other assessments, those gold standards for every therapeutic indication, whether it's the positive and negative symptoms of schizophrenia for schizophrenia studies, or whether it's an ADAS-Cog for Alzheimer's, or whether it's a WOMAC for a pain, or whether it's a dermatology-related scale for dermatology-related studies. You're getting the raw data, the patient, how they are baseline before being assigned the investigational product. So you're going through everything again. You're going through most likely all the same assessments you did at screening, say everything, labs, ECGs, all that stuff, and you're going through the inclusion-exclusion criteria again, because you get one more chance to not improperly enroll somebody into a study. Okay, now, you won't get baseline lab results back the same day. So you're gonna, usually inclusion-exclusion criterias don't have a baseline lab exclusions. Unless it's a urine drug screen or something you can do on site. So, but there will be exclusions for some of the other assessments like EKG. There will be exclusions for some of those gold standard of assessments for the particular therapeutic area. Oftentimes they have to rate or assess or have outcomes similar to their screening outcomes in order for them to still be eligible. So that's probably the most complicated part of the baseline visit, is to ensure that yes, they indeed do qualify. If they do, if all the inclusion-exclusion criteria are met, and I do mean all of it, every single last one, then you can go in the IRT as a coordinator with your PI's approval, of course, and if any doubts, with medical monitor approval, and you can hit randomize. You can hit the randomize button or the enroll button, whatever your IRT has designed for that study. And then it usually spits out, the IRT spits out the IP number that will be assigned to that patient. Whether it's double blind, whether it's open label, whatever, there's always an IP associated with randomization. Well, always, or for the most part. So I'm trying to keep this simple and under 10 minutes. So that's what you do. Then you go get the investigational product. Make sure you understand and look through your SIV training slides, look through your protocol, call your monitor if you have to. The first randomization you do for every study is going to be your, unfortunately there's no way around it, you're going to learn a lot. All right, you're going to learn the traps of the study. If you're going to make a deviation, you're probably going to make it early on in the study. This is why monitors like to come out within 2 weeks of your first randomization. So just be prepared, be extra careful. They get easier as you start doing more, but the first ones, just like the first screening visits, the first randomization visits, go slow. You're going to try to avoid as many pitfalls as you can. You're going to grab the investigational product. You're going to know how to properly instruct the patient on how to dose. You're going to know how to properly instruct the patient on any other things they've got to do, such as patient-reported outcomes, right? Sponsors love those now, so you got to properly train your patient on how they're going to enroll or how they're going to enter data in the electronic patient-reported outcomes, or what they need to do. For example, when are they allowed to eat with taking the IP? What times should they take the IP? What do they need to record? Do they need to record their adverse events? The answer is yes. In the ePro, do they need to notify you of any adverse events if there is no ePro? Yes. Do they need to take the IP at a certain time? Do they need to take it with food? Are they allowed to shower? All these things you need to know. so you can tell the patient, because you have no excuse. If the patient's not compliant, it's your fault as the coordinator, because you're supposed to educate them on this. And yes, easier said than done. We're busy as coordinators. We've got a lot going on, but it gets easier. Just figure out the right process for your site to be compliant. Keep in mind patient safety at all times, and you're good. Okay, so that's it. You assign the IP, you make sure that they know what they're supposed to do, whether it's patient-reported outcomes or anything else, and you make sure that they understand they can call you at any time with any questions, report all adverse events to you, and you schedule their next visit, which should be based on the protocol, and that will be usually visit 3. And from then on, they no longer have funny names. The visits no longer have funny names. They are just visit 3, 4, 5, 6, 7, and so on. So hopefully that helps. In a nutshell, what is a baseline? What is a randomization? Again, we didn't even get into double blind. Double blind basically means you don't know, you the site, you the PI, and the patient, you all don't know what IP was assigned, if there is some to be assigned. If it's open label, then obviously you know what you're assigning. You may just have a variable on the dose, which could also be open label. So that would be called open label, so unblind. And there's also single dose, so then everybody knows what they're getting. And sometimes it's single blind, meaning just the patient doesn't know what they're receiving, but the site does. So that's the only differences. Don't get too confused on that. If you need more help with that, watch my 5-hour video where I go through all of this in greater detail, but I'm just trying to keep baseline / randomization visits short and sweet. Like, subscribe, comment, share. Thank you so much, Guru Nation. Bye-bye.